we're here this evening with a very limited group in person, but we're happy that people are coming out in person, and we offered it hybrid so people can view online. My name is Carrie Picani, and I'm the president, re-elected for the, I don't know how many years, but I also wanted to announce tonight our new officers that were elected. We did that this fall. We do that every fall, and so who's here now is Susan. She's our vice president, plant sale manager, venue coordinator, and whatever, a ton of things. And then we have Michael here tonight, and he's our new secretary. And also here tonight is Carolyn, who is our outgoing treasurer, but she's still involved in many things, and we're very grateful for that. And then for those of you viewing from home, we have Ray Corbin and Barbara Erder as our members at large. We also have Don Essig as our treasurer, Kirsten as our membership,
Vicky, the
voice in the cloud, who's our state. She's our state membership, and she also helps us out tremendously with our Kobe chapter, and she coordinates our Zoom presentations along with Kirsten. I hope I'm not forgetting anyone. Okay, all right, so I think I have everyone down, and Peggy also has been helping us with education and outreach. So we wanted to thank those individuals who help us keep Pahobie going. Our next presentation after Megan's, which will be on December 13th, is Rob Laporte. Hopefully I'm saying his name correctly, with the College of Idaho. He's the new director there. And then we should be receiving a sage note that will come out in December. And then reminder to renew your membership for those of you who may have not yet, but it's end of the year is usually when membership dues are due.
And notice?
We will be sending out a notice to chapter members, yes. All righty. Okay, so I think that's all I have for announcements.
Could I do an announcement about
yes.
the bus trip?
Yes.
Bus trip.
I guess I can just do it from here.
We can turn it.
Okay.
Yeah.
Do you see
We
our
are
Vicky?
hoping to offer a bus trip to Orton Botanical Garden in next May. We did one 4 years ago and people really liked it. And Kevin Laughlin suggested that we really needed to do another one when he and I were at Mary McGowan's memorial service. And so I've inquired, and the Botanical Garden would be happy to host it. And the only thing is we need to fill up the bus in order to pay for it. It usually costs about, each person pays about $25, I think, but bus prices may have gone up considerably then over the 4 years. Anyway, Carrie will keep you posted every now and then, and you'll get reminders and sign up for stuff. But it won't be till May.
Anything else? Any other announcements? All right. So I'm very happy to welcome Dr. Megan Cattau, who is an assistant professor in human environment systems at BSU, or Boise State University. She received a Bachelor of Fine Arts in Painting at the University of Georgia. I did not know this. I also have our
Cool.
a Master of Environmental Management from Duke University, and a PhD in Ecology, Evolution, and Environmental Biology from Columbia University. She worked as a research scientist at the Earth Lab, University of Colorado Boulder. She specializes now in using remote sensing and geospatial analysis to answer theory and management relevant questions about ecological disturbance. She focuses in particular on the human contributions to fire disturbance and recovery dynamics of social ecological systems. She is an advocate for making environmental science more accessible through inclusive
Pedagogical, education, inclusive education. Yeah.
approaches, community and practitioner involvement in research, and collaborations with artists that bring science to the public. And we are very grateful to have her here tonight, as she is our first in-person presenter since pre-COVID. And thank you very much. I'm turning it over to you.
Thanks.
Okay, and then I'll turn the camera.
All right, thank you. As Carrie mentioned, I'm with a group called Human Environment Systems here at Boise State. I want to thank Carrie in particular for inviting me to give this talk. Also, you all for joining me, particularly since we needed to reschedule. You can see I optimistically have November 8th on my slide still, which is the original date of this. Glad to have you all over Zoom, who are over Zoom. I'm excited to have a chance to speak with people who are so passionate about and knowledgeable about native plants, which is a little outside my area of expertise. But glad to be here. And I'll be discussing mapping of invasive species, which I know is kind of a controversial term, but I'll be using the term invasive species throughout the talk, but we can chat about some maybe more appropriate terms if you feel strongly about that. I'm always glad to have that conversation. And mapping them using drones. And I'll say unoccupied aerial systems or UAS, and that just means drones. Those words are kind of interchangeable. I'd love for this to be relatively informal, so feel free to ask questions at any point. Stop me if you have a clarification question, or if you have a follow-up, even comment, and there should be some time at the end of the talk too. Those of you on Zoom, I won't be able to see the chat, so maybe you could just yell into your microphone if you have a question that you want me to answer as I go along. So I want to give just some brief big picture context here. You'll all be pretty familiar with this. I just wanted to mention it as a way to talk about the framing and sort of the motivation for this work. I see the lighting is changing. This work takes place within the sagebrush steppe ecoregion of the Great Basin, which is shrub steppe, dominated by mostly perennial bunch grasses and sagebrush shrubs. And sagebrush is estimated to have covered about 150 million acres in Western North America, sort of historically. And today, this ecosystem type covers about half of its original extent. And it's estimated that less than 10% has retained its historical structure and composition. So the extent has shrunk, and even more so, the makeup of that ecosystem has been altered pretty considerably. And as you all know, likely, invasive species are one of the most prominent threats to native vegetation in the Great Basin. And climate change has really been the catalyzing force of invasive species establishment and spread, and has worked in tandem with changing fire regimes in this area. And that's true in the area around Boise as well. Warming and drying conditions sort of combined with increasing human ignitions have increased the length of the fire season, and fires also becoming more frequent and larger, and in some places sort of more severe. And that's true across the US West in general, and including in sagebrush steppe ecosystems. So these changing fire regimes interact with invasive plants that can outcompete native vegetation. Many of these species deplete soil moisture earlier than native vegetation, and so they suppress native species growth. And when these fire regimes, or when these invasive plants alter fire regimes in ways that promote their own persistence, a feedback loop is formed. So these species, for example, can shorten time between fire. And so this annual, invasive fire cycle develops in that way. And the fires will then burn too frequently for native perennial vegetation to persist in some cases. So also in relation to climate change, we see rising atmospheric carbon dioxide concentrations. This elevated CO2 will likely increase plant biomass and thus fuel loads kind of generally. But there are two issues that are really specific to invasive species related to the elevated atmospheric CO2. And one is that we have enhanced wildfire intensity with that increased productivity, just because there's more fuel, right? More potential fuel loads, and so more intense wildfires. And the second piece is that cheatgrass, which is a common invasive species around here, actually responds more strongly to CO2 enrichment than native perennial grasses. And so, then the perennial grasses and also forbs really. So that means that the biomass increases disproportionately for cheatgrass, and again, increasing fuel loads. So you can imagine how this fire invasive cycle works, especially in the context of increasing CO2. Historic fire return intervals, or the time between subsequent fires in this area, are estimated between 30 and 100 plus years, which is a pretty big range, right? But invasive annual grasses, where those have become established, fire return intervals have become shorter, and some areas have experienced wildfire every 5 to 15 years where those invasives have been established. So even though it's not completely certain what the historical fire regime would have been in this area, the range is big, 30 to 100 years. we know 5 to 15 years is much shorter, right, than what we would have seen here historically. So post-fire recovery and sort of current management practices just can't keep up with that. So because of these factors, exotic annual grasses have replaced native species in a lot of areas, especially in the low to mid elevation sagebrush steppe, where you see less resilience than in the upper elevations. Okay, detecting and mapping invasive species is a requisite first step toward understanding changes in their distribution and abundance, right? If we want to manage the spread of invasives effectively, it's important to understand where they're even located. We could go out into the field and do a survey, which is a common method of understanding where invasive species are more generally, but it's resource intensive. You know, it takes a long time to look on the ground, on foot for specific species. A lot of you have probably done that work, so you know how time intensive it is. And because of that, field-based studies are often quite limited in spatial extent. We can only cover so much area on foot, given the person power and the time that we have available, right? So what our group wanted to know is, can we use remote sensing technology to detect and map invasive species over larger spatial extents? And I want to talk just briefly about what remote sensing is. And again, this is a place where you can feel free to interrupt me if you have a question. So remote sensing in general is just about detecting an object without having contact with it. That's sort of the most basic definition of remote sensing. Anytime you're measuring something and you're not touching it, technically that is remote sensing. And it can be from any distance. I think we most often think of satellites when we think about remote sensing. I mean, is that true? Do you all think of satellites when you hear the term remote sensing? Most of you are nodding. But remote sensing can happen on a variety of platforms, right? It could be satellites, which is what you see in that graphic on the very top there. It could be manned aircraft or personed aircraft, including propeller planes. It could be unpersoned aircraft, including drones. It could even be like a handheld object that doesn't actually come into contact with the object of interest. Technically, that's remote sensing too. And we're going to be focusing mostly on drone-based or UAS-based remote sensing in this talk. But this is generally how remote sensing works, specifically passive remote sensing. This is using a satellite as an example, but this is true for any remotely sensed platform that's measuring light. So solar radiation, the electromagnetic spectrum, comes in from the sun, it passes through the atmosphere, it interacts with objects on the Earth's surface. So plants, in addition to things like buildings and water and roads, everything that's on the surface. That the light that's reflected is then measured by the remote sensing sensor. And in the case of satellites, that light has to go back through the atmosphere. In the case of drones, it's not having to go back through the atmosphere to be measured. But in general, that's the kind of remote sensing that we'll be talking about today, is the kind that's measuring how light from the sun is reflected off of different objects, just sort of very basically. And I just want to mention that the visible part of the spectrum, or the part that we can see, is only really a very small fraction of the entire spectrum. So this is all the light that's coming in from the sun. You can see that very narrow piece of the spectrum is what we can see, right? From purple all the way through to red. But a lot of remote sensing platforms also will let us capture ultraviolet and infrared and near infrared, sometimes into radar. So you can imagine remote sensing can offer us a lot of additional information than what we can just see visibly, given how much of the spectrum is not visible to us. This is how light interacts with elements on the ground. So again, that light is coming in from the sun. Some of that light is absorbed, some of it is transmitted through objects, and some of it's reflected. And the piece that we are measuring is the piece that's reflected. So you can see that this plant is green in this example. So that plant is reflecting a lot of green light, right? But most healthy vegetation reflects a lot more in the near infrared part of the spectrum than it actually does in the green part of the spectrum. So if we could see near infrared, vegetation would look near infrared to us, which I think is kind of cool. So I think for vegetation sensing in particular, that highlights the benefit of having sensors that can measure outside of that visible part of the spectrum, because that's where the most powerful signal is coming from plants, is mostly in the near infrared. This graphic is just driving that point home. So hopefully you all on Zoom can see my cursor. But if we just take a look at this green line that represents green vegetation, on the x-axis here is the different parts of the spectrum. So the visible part of the spectrum is right in here, and then it moves into near infrared and mid infrared. The y is just how much, the percent reflectance. So how much of that light is being reflected off an object. So this is called reflectance spectra. The green line here is green vegetation. This little peak right here is the green part of the spectrum. So that's the amount of light that's being reflected off of plants that we see only really is green, you know. This is near infrared. So that's how much more near infrared is being reflected from green. And this is a generalization. This is not every single plant, of course, but this is sort of generally green plants are reflecting a lot more in the near infrared than they would be in the green. And then kind of interestingly, if we compare with dry vegetation, which is this kind of brownish reddish line, you can see in the green part of the spectrum, it's almost the same percent reflectance, right? So dry vegetation might even look green to us, but look at the difference. Oh, my cursor's gone. Look at the difference in the near infrared. There's a huge difference, right? In how much near infrared dry vegetation might be reflecting relative to green vegetation. So again, there are things in using these sensors that are outside of the visible range. There are differences between plants that we can detect that we can't even necessarily see with our eyes. Okay, the use of remote sensing technology to detect and map invasive species over large spatial extents is limited in part by the spectral similarity of different species. So for example, different species might just appear as green or might just appear as near infrared at a given time of year. This is an example from our field site. You can see Squirreltail on the left, which is a native bunch grass, and Cheatgrass, an invasive annual, both appearing as sort of relatively green during one of our field days. Green for this ecosystem anyway. In the tropics, this would not be considered green, but so you can, obviously when you go to the field, you can see very clearly the difference between those two things. You can, just from the structure, the morphology of the plants. But if you're thinking about a remote sensing platform, it's just looking at the light reflectance. And so on that remote sensing platform, these things might look identical in a given time of year, right? But the differences in phenology, or the timing of the growing cycle between species, can result in these unique spectral signatures over time. And that would enable us to differentiate these species from one another. And I'll talk a little bit more about what I mean by that. But so some invasive species, as you know, may green up or brown down earlier or later in the season than many native types of grasses, allowing for possible detection. So for example, we know that in some sites, at least, cheatgrass often starts growing earlier in the season than some native grasses and dies earlier as well. And that's not true universally, but it's true in a lot of places. So if we take a series of remotely sensed images over time, we could differentiate those species from one another based on when a given pixel is greening up and when it's browning down. So the timing of that phenology allows us to understand what species it is. Any questions so far at all? Okay, great. So the use of this multi-temporal data or multiple time series data using satellite has been around for over a decade. So people have tried to leverage that phenology using satellites for a long time. So, excuse me. For example, in this ecotype, Singh and Glenn use a time series of Landsat data to produce this map of cheatgrass in the sagebrush steppe. So this is using a really commonly used satellite called Landsat, and each of those pixels is 30 meters by 30 meters. So for 30 meters by 30 meters, you get one reflectance value for near infrared, one reflectance value for green. So it's very coarse, right? And so there's another study by Spate et al., and they use a different satellite, which is RapidEye, and it's a 7-meter resolution. So it's, compared to the 30, 7 meters is quite a bit smaller. And they did a time series to map exotic annuals. Both of these studies found the species of interest really difficult to detect at low cover thresholds. So you can imagine if you have a 30-meter pixel and even just 30% of that is occupied by invasive species, you might totally miss that species, right? It's only when you have a really homogeneous, spectrally pure pixel that it's easy to detect those individual species.
What are the black dots?
The black dots there, I think, are their sample sites. Okay. Yeah. I wasn't involved in this paper, so yeah. This is just the example using Landsat to show cheatgrass distribution in the area. So approaches using satellite data in general, including in these two studies, are limited by the relatively coarse pixel resolution, 30 meters and 7 meters in this case, and also by satellite return intervals. So Landsat, that one that's commonly used, visits the same place every 16 days. So there's a lot of change that can happen over that 2+ weeks that you might miss, right? So we think these limitations can be overcome by incorporating high resolution drone data or UAS data. UAS is a particularly suitable tool to detect species-specific phenology because of the flexibility in data acquisition, meaning you can go out anytime you want. You could fly every day if you wanted. You could fly multiple days if you wanted, if you really need to get a finely resolved phenological curve. Also, there's some flexibility in flight planning. So depending on how high you fly, what your sensor is, you as the user determine what the pixel size is. So your pixels can be as small as a centimeter, very fine resolution. Data can be, again, collected on demand at a sufficiently fine spatial grain to capture spectrally pure pixels. So you can imagine if your pixel is a centimeter by a centimeter, that's going to be an individual plant that you're detecting. So you can really get the signal pretty strongly there. So even in communities where there's really diverse vegetation that are spectrally mixed, drone data might be able to disentangle those plants. Just to drive home this point about scale, the image on the right is about 140 pixels squared from our multispectral drone imagery. So this is 5 centimeter pixels. And again, that was the scale of that rapid eye satellite that I mentioned they had used in a previous study. So you can see how much variation there is among these different drone pixels. So this image is a bunch of drone pixels, right? And the rapid eye, this would be one single pixel. So you get one single value for that whole area. Whereas here we're getting, thousands of different values. So you can imagine the scale of Landsat, which is 30 meters, would even be coarser, right, than this 7 meters. So we want to know, can we leverage the capabilities of drones to collect fine scale data and also the unique timing of the growing cycle between species to detect and map these species? That's our general premise and our interest here. It's not advancing, sorry. I'm getting a spinning wheel. So that's not ideal. So we'll see how this goes. Oh, great. Okay. So I'm just going to play a little video. Sorry, it's a little dorky, but this was a team that came out to capture some of this work. This is part of a bigger project, and they were capturing work in that whole project. And I think they did a good job of sort of synthesizing what we're doing and getting some nice imagery. So hopefully this will work. If not, I apologize, but. I give it just a few more seconds and then we'll move on if it's not happening. Okay, it's not working. I'll try to play it at the end. Oops. Oh, sorry. Spinning wheel again. Okay. Sorry, everyone, it's just a little frozen here. There we go.
My overall research is about understanding how changing disturbances are affecting our landscapes and are impacting social ecological resilience. This project is about using the phenology of species or seasonal changes in the growing cycle to detect. So this project is in the pilot phase. We come out every 2 weeks to this point here, which is initial point, with a multispectral sensor mounted on a drone. We first arrive at the site. We put out ground control points, which are points that allow us to later in the lab tie the imagery that we take with the drone to geographic space or physical space. After that, we calibrate our drone equipment And then we fly our drone along a pre-programmed flight path that allows us to take data consistently week after week. We can't combat invasive species if we don't know where they're located. So this project aims to understand how can we use the unique type of seasonal growth cycles of individual species to identify them and to map them in drone imagery. And this has far-reaching applications, from research applications in which we might look at interactions between invasives and local plants, or between invasives and bioprocesses, as well as management applications in which this information can be used to help for prioritization of mitigation efforts of invasives. Some of the species have evolutionary strategies that give them a competitive advantage. So for example, cheatgrass, because it grades up earlier in the season, the native vegetation, it's ready to send its shoots up. Well, a lot of the native grasses around to their root systems, putting that cheatgrass at an advantage. And we are using those very evolutionary strategies that give those plants an advantage to find them, to detect them, and ultimately to help control them. And I think that's really neat.
Okay, hopefully we'll be able to exit out of this. Okay, great. Also, you probably know that you may have noticed in that video it was an all-female field team, which was often the case at that site, and I thought that was kind of fun also. One of those people is Anna Roser, who you know. So you saw our site was at Initial Point, which is near Kuna, Idaho, and I want to talk just for a moment about how we selected that site. So we took a collaborative approach whereby, we had interviews with a variety of land managers, and that included Zoe Duran from the Idaho Army National Guard, Anne-Marie Raimondi and Joe Weldon from the BLM, and Martha Brayback from the city of Boise. So we asked what species and what areas might be of particular management interest. And based on those conversations, we landed on this site because of its location and because of its species composition. The site's within the Snake River Birds of Prey National Conservation Area, which is important for nesting and wintering raptors, including things like prairie falcon, which makes it a valuable area from my perspective, but also presented its own kind of suite of complications about just making sure we're being very careful with where we were flying the drones, not close to nesting sites, of course, right, that type of thing. So this is arid sagebrush steppe system. It's a relatively degraded site. The species we examined at the site, it did include some native grass species, including sandberg, bluegrass, and squirreltail. There were also planted vegetation species, including Forage Kochia and Russian wildrye. These species, these planted ones, were likely here for the same reason we see crested wheatgrass, right, all across this region. So they were planted there with the idea they could serve as a bridge species that would occupy a depleted site to prevent further degradation and limit exotic annuals, but allow transition to a native dominated plant community sort of eventually, right, is the idea behind that. These species are still frequently used, crested wheatgrass in particular. They're frequently seeded after wildfires because of this presumed ability to suppress exotic annual grasses, because it's relatively low cost to plant them and they establish pretty easily relative to native species. After fires, agencies will often drill seed this Russian wildrye, Forage Kochia, and crested wheatgrass. The efficacy of this approach is pretty hotly debated, as some of you are probably familiar, but I see some nodding. But these species are there, and that's probably why. The site is largely dominated by invasive annuals. So mostly Russian thistle. That was a lot of the cover. Summer annual, Burr buttercup, the winter annual, for which we saw this pattern from small leaves to thick mat to dry spiky seeds over the season, and cheatgrass, which is a winter annual primarily. And in this site, it was largely limited to the roadside, so it wasn't very distributed across the site. We also took note of some other background land cover types like bare soil, rock, and moss, and some biocrust out there too. So we conducted repeat drone flights to capture vegetation over time of these different native, planted, and invasive species. So we used 2 different types of sensors, and I brought those here today, so you might have an opportunity to look at them. You may not find this interesting. I think they're kind of interesting just to see. The first is this multispectral sensor, and there's also a picture of it up on the slide. So if you can't see this very well on the camera, you can just look at it up on the slide, but I wanted to give you a sense of the scale. So this is a multispectral sensor. Each of these sensors is capturing a different part of the electromagnetic spectrum. So red, near infrared, blue, green, different parts of the spectrum. This captures, spectral information, which is that light reflectance that we've been talking about. And we'll focus mostly on the data from this sensor tonight. And this sensor is mounted on the drone, typically, that you can see in front of us. Sorry, those of you on Zoom, it's the same drone that's in the picture on the bottom left and the bottom right. But you can see it's, those of you in person at least, it's relatively big, but manageable, right? So you can fly a pretty sophisticated sensor on a relatively small drone, which is neat. I mean, 5-10 years ago, that would not have been possible.
How much does that weigh?
This one? I am not sure. I'm going to guess 5 to 10 pounds, like pretty light. Who asked that? Oh, yeah. You can pick it up later if you're interested. But one person can, it's a little awkward, but one person can handle it pretty easily. So it's really heavy in the case and everything, but if necessary, one person in the field can totally manage that drone. We also flew an RGB camera, a red, green, blue camera. This is just like your standard phone camera. Excuse me, that sensor is actually better for helping us capture structural information or creating 3D visualizations of the landscape later. And that's this. It doesn't have the propellers on it right now. This is a sensor. I mean, you can see this is very light, right? And even in the case, it's easy to transport. So we take the RGB data and turn it into 3D reconstructions using a process called structure for motion or photogrammetry. We won't get too much into that type of data this evening, but happy to chat with anyone about that type of data later too. Okay, we completed 12 flights distributed approximately every 2 weeks from March to November with that 10-band multispectral sensor that I just showed you. And the data we collected is at a 5-centimeter resolution, so pretty small pixels. This is just an example of what that imagery looks like, that figure that you can see on the slide there. Here we have the green band, so only the data coming from the green sensor is displayed. You can clearly see the peak green up for Burr buttercup in late March on the figure on the left, and then how most of the vegetation has senesced by midsummer in July in the figure on the right. Okay, so here we're seeing NDVI over time. NDVI is just a metric or an index of vegetation health. It uses the near infrared and the red bands to approximate plant health. So when it's green or when the value is 1, you can think of that as healthy vegetation. And when the value is brown or closer to 0, that would be senesced vegetation. So with this NDVI time series animation, the first pulse of green, I'll wait till it gets there, is once again that Burr buttercup. So that's what we see kind of there. So that's the Burr buttercup, and that rapidly browns down. Next, some of the bunch grasses green up with the larger Russian wildrye showing up well in the imagery. And then late in the year, those pockets of green are Russian thistle and Forage Kochia. So you can kind of get a sense of what this spectral information over time looks like in this time series. Okay, this is the same information that was embedded in that time series animation. It's just NDVI graphed over time. So on the x-axis here, in case you can't read that, that's just date. So it would be, probably about March, April, all the way through October. And this is, again, that index of plant health. So the higher the number, the more kind of healthy the plant or the greener the plant in that moment. The colors here are representing different species or land cover classes, and the dashing represents native, invasive, or planted. So for each species, those lines represent the different phenological curves or phenological changes over time. The native species are solid. I'm not sure if you can, those of you on Zoom probably can tell, but in person, I'm not sure if you can distinguish the dashing, but the native species are the solid lines, the invasive species are the short dashes, and the planted species are those longer dashes. And this black line at the bottom, let's see, get my cursor over there, is a background soil value. This one is sort of the background soil value.
What is POSE?
POSE. So yeah, Poa secunda. Yeah. So this, I'm going to just show you two examples. This bright green dotted line is Burr buttercup, which is an invasive winter annual. So you can see it reaches its peak value or its greenest value pretty early in the season. And you saw that in that NDVI animation too, right? That hot pulse of green at the very beginning was Burr buttercup. And then it senesces pretty rapidly, right? So in April, it starts to senesce pretty quickly. If you compare that with another example, the light blue solid line, which is squirreltail, native bunch grass. So this one. It has its peak value just a little bit later in the season relative to the Burr buttercup, not much later, right? But it has a lower magnitude of NDVI. Oops. So you can see this difference, right? It's just a slightly lower magnitude. And if you were to look at both of those species in late July, for example, they might look identical to one another, right? Like the lines are touching each other. So when I showed you, I can't remember what the two species I used as an example, two species sort of looking identical in the field, that would be the moment, sort of late July, if you went out into the field, those two species would look identical to one another. But you can imagine how we could use earlier in the season, there's some really clear distinctions between those two species, and we can use that to differentiate those species from one another. Here's another example, this dark blue solid line here. Again, that's the Poa secunda that you were asking about, this one right here. Or Sandberg bluegrass, it's native bunch grass. It also has its peak value early, but at a much lower magnitude relative to the Burr buttercup. So if you were to look at both of those in like April, where those lines cross, they might appear identical. But again, looking at a time of year in which there's some distinction can help you differentiate them. So here I'm just pulling out the 3 invasive species at our site, which have really distinct peak green up and senescence states. We were really surprised about how distinct these were from one another. So we have Burr buttercup in green, cheatgrass in that reddish crimson color, and then Russian thistle in that magenta color. So we see that nearly every species, including these 3 species at our site, is uniquely distinguishable based on the date of peak NDVI. So when we see that peak, and also the magnitude or how tall that peak is. So again, that's our peak dates there. I'm just toggling back to all the species, just so that you can kind of visualize that. So we have 2 different ways of assessing accuracy. We used a machine learning algorithm called support vector machine classification, basically to take every pixel in our drone imagery and assign that pixel a species. So that's a classification in general, is categorizing all of your remotely sensed data, in this case, into species. And so here we have an over 90% accuracy for those samples. So using phenology to classify species in this drone imagery turned out to be a really effective strategy for classifying species, which was exciting to us. So before I move into sort of next steps, I wanted to ask if anyone had any sort of questions about, yeah.
Did you ground truth?
Yes, yeah, I should have mentioned that. So we have high precision GPS, and we took at least 50 points for every species, which is how we created those original phenological curves. So we knew really clearly what species belonged to a given pixel, then we could track that pixel over time. And then we use that information to classify the rest of the pixels. Yeah, it's a good point. It's really important to ground truth your remotely sensed data, because you can sort of speculate looking at it, but unless you've been on the ground and verified with your eyes, it's you could be wrong, right? Just looking at the drone imagery. Yeah.
Do you think the site is unique and that the NDVI is able to separate the species out? Have you tested the other areas in southwestern Idaho to see if there's more overlap?
Yeah.
Clear distinction?
That is also a great question. That is in our next steps. I think the site is not unique in that these species are distinguishable, but I do think it's unique in that there are a lot of factors that are going to affect the unique phenological timing of each of these species. So we might see shifts earlier or later, just for example, as you move up and down in elevation. But I am not surprised, and I think that the curves will be unique at other sites. They'll just look different than the curves at this site. Yeah, that's a really good question. Yeah.
How big are your pixels again?
5 centimeters.
5 centimeters.
Mhm.
So you can be relatively confident that there's one species per pixel.
Yeah.
more or less.
Yeah,
So your
absolutely.
GPS or your ground truthing can tell you exactly what pixel you're in. That's
It's
5
like,
centimeters?
it's centimeter precision. Yeah. I know. Yeah. Okay. So the primary objective of our next steps is just to improve the classification, right? So the classification algorithm performed really well, over 90% accuracy, but we're testing ways to improve it further. As I mentioned, we have this RGB data. And so we have structural information, like for example, plant height across the site that we can use. And we took this structural data, we took the RGB data at 3 distinct times in the growing season. So we don't have it every 2 weeks, but we do have 3 different time snaps of the structure. And that's at 1 centimeter resolution. So we're incorporating, we've built the structure for motion or the photogrammetry models so that we have generated those 3D products of the site. And that could help us differentiate vegetation with similar phenology, but perhaps different size or shapes. So one example of that is Forage Kochia and Russian Thistle. Those green up at kind of a similar time, and they both had variable senescence dates, but the shape and structure of those are going to be different. So that could incorporate that information could improve the classification even further. We're also working with those aforementioned agency partners and land managers to select additional sites, as kind of as your question alluded to. The pilot site was on BLM managed land, but we've included ecologists from the Idaho Army National Guard and City of Boise with hopes to expand into those management areas. And I'm open. If people know of other sites that are interesting from a species or a location of management perspective, I'd love to hear anyone's input on other areas you think would be good to fly. So in kind of working with these people, we can target sites of particular management concern and or different species. And an example of this is that tumble mustard is another species of concern that we have not yet been able to capture. So ideally, some of our future sites would have tumble mustard in them. And we'll also be able to, again, this is kind of speaking to your question about other sites, we'll be able to cover a range of variables that would affect those phenological patterns. And that would be biophysical variables as well as climatic variables. So things like elevation, topography, species assemblages, those are going to help us develop basically biological or biophysical correlates of phenological patterns over these larger spatial extents. So this is the site that we picked was a relatively low elevation site. If we move up in elevation, for example, we might see that green up shift to later in the year. This is just an example of how some of these factors might affect the timing of those curves.
Who was the land manager in Kuna?
The one around Kuna was Zoe Duran is who we worked with for that one, the Idaho Army National Guard. Yeah. And then, this point is related to the previous one, which is that, given this workflow that I described, it's relatively resource intensive. It's not as resource intensive as going out and collecting field data, but it does still take some person power to go out and collect the drone data. We're really interested in scaling this work up to satellite data. And those correlates of growth cycle timing that we're hoping to test would enable us to do that. Okay, so in general, this work fits into the growing body of work that's testing the use of UAS approaches to improve mapping of ecological resources, just more generally. Our first primary takeaway here is that resolving phenological patterns for our species of interest, at least at this site, does improve our ability to detect and map them. So that we found that very encouraging. So from a method standpoint, the UAS-based multispectral time series data were effective for differentiating species, including native species and invasive species at our site. And interestingly, I thought classification with spectral data alone was successful. Again, just using the date and magnitude of peak NDVI. I thought for sure we would need to have that structural information incorporated in there for this to work, but just the spectral data alone was effective. I was really surprised by that. So these methods using phenological differences in species to differentiate in UAS imagery can likely be applied more broadly, really in any system where the invasive species have different phenology than the native species. You could use a really similar approach or methodology to detect and map those species also. And our second primary takeaway is that these efforts to resolve the phenology will hopefully have benefits for research and management. Hopefully will contribute information towards spatial prioritization of management efforts that do require that spatially explicit knowledge of species occurrence. Also, something that we haven't delved into too deeply in our own lab, but there are a lot of mitigation efforts that leverage phenological information. So for example, you might apply post-emergent herbicide during a specific life stage of a plant. And so knowing the unique phenology of those species at a site could help you target when those life cycle specific mitigation efforts are enacted. And so from the research side, this project hopefully will contribute toward modeling efforts of, for example, interactions with native species, including sagebrush, and interactions with disturbances like wildfire. I just want to acknowledge partners at Boise State, as well as our agency and land manager partners. And yeah, looking forward to expanding the project as previously discussed, both in order to capture additional areas of management concern, as well as to capture those correlates of the phenological patterns. Thank you all so much for being here in person or over Zoom and listening to me geek out about drone imagery. I'm so excited about the prospects. So yeah.
Thank you.
Yeah, thanks. Yeah.